Imagine a future where we could stop type 1 diabetes from progressing, so someone never has to start using insulin. Some therapies already exist to delay onset, but what if we could stop progression or delay T1D indefinitely? One company believes it is on the path to do just that, and today they're on the show to tell us more. Welcome to Diabetech. I'm Justin. I have type 1 diabetes, and on here I talk all things diabetes technology, research, and innovation. This is my weekly podcast that airs on Mondays, but I've got more videos on YouTube on Fridays and a blog that puts out articles every week. That is diabetech.info. Links to that and my socials are in the show notes. SAB Bio has developed a treatment that could delay or prevent the onset of type 1 diabetes using a human version of thymoglobulin. Traditional thymoglobulin is derived from rabbits. It's used in transplant medicine and has shown promise in preserving beta cells in type 1 diabetes. But because it comes from an animal, it can cause a reaction known as serum sickness. SAB Bio's approach eliminates this issue, potentially making it a safer and more effective treatment. The company is advancing into its phase 2B clinical trial for its therapy SAB-142. Today, I'm joined by Sam Reich, CEO of SAB Bio, and Dr. Mike Haller, a leading expert in diabetes research who has worked with similar therapies, including studies on Teplizumab, which delays the progression of T1D. We discuss how this new therapy builds on past research, why it could be a game changer compared to existing treatments, and what its latest clinical trials are revealing. Plus, we dive into the science behind the therapy, including how it's developed using genetically engineered cows, and what it could mean for those newly diagnosed or at risk for type 1 diabetes. Keep in mind that this podcast is for educational purposes only, not medical advice. Always consult with your physician before making changes to your healthcare. Also, I am a spokesperson for Tzield, which comes up in this episode. That relationship has had no effect on my reporting or the questions that I ask. Let's get into it. Welcome to the show, Sam and Mike. It's great to have you here. I'm looking forward to getting into all the research and work that you've done in the space. I have done my absolute best to organize this interview in a way that I think makes most sense, but there's so many things we're going to talk about that just I don't fully understand. So I'm going to ask you some pretty simple questions here and there, just so me and my audience can really understand everything that's going on. And if I'm hoping that we don't hop around too much based on my outline, but I think it's going to be a great conversation either way. So it's good to have you here. First, I want to kind of introduce who you are and how you're related to all this. So Sam, let's start with you. Tell us a little bit about your background and how you arrived at SAB Bio.
Sure. So in college, I majored in molecular biology, and I went to the University of Pennsylvania to do a PhD in cell and molecular biology, which is broken up into a lot of subsections, and I was in the gene therapy training group. And I got to train under some amazing innovators in gene therapy and therapeutics. My mentor invented the first gene therapy to be approved, which is called Luxturna. And when I was in grad school, I made some inventions for new therapeutic modalities. I started a company, it was called Acuity Pharmaceuticals, and got into the entrepreneurial side of biotech and began sort of developing drugs outside of the academic lab and in a more commercial setting, which is where venture capitalists come in and fundraising, and then really getting into product development rather than exploratory discovery research. This is my third venture. That was based on siRNA, and I was working in ophthalmology. siRNA was a therapeutic modality that I was involved in inventing in the early zeros. And there are several approved drugs that use that approach. And then I got involved in epilepsy with an epileptologist at Harvard. And he invented a drug which is now working its way to approval. And, we brought that out of Harvard and brought it into the clinic and then brought it to a bigger pharmaceutical company who took it over. So I have a scientific background that has turned into a bio-entrepreneurial background which really involves taking an exciting discovery from a lab at a university like Dr. Mike Haller's lab and raising money in order to validate that that technology is going to be a drug that helps a lot of patients that need help, need some new innovation, some new product. And so for the past 3.5 years, I've been working at SAB to bring this exciting new technology forward in diabetes, and closely collaborating with Mike, who's I think was one of the originators of the idea that this would be a good approach and has generated a lot of great data to show that what we're doing and what you're about to hear more about has tremendous promise to help patients with type 1 diabetes.
Yeah, Mike, why don't you tell us a little bit about your background and how you got involved with SAB?
Sure, thanks for having me on. So I'm Mike Haller. I'm a pediatric endocrinologist. Born and raised in Gainesville, Florida. I was a science-interested kid. My grandfather had type 1 diabetes, so that was always a side interest. But knew early on I wanted to go into medicine. Went to Duke for undergraduate, and then came back to the University of Florida, where I did my medical school training, residency in pediatrics, fellowship in endocrinology. And along the entire way, continued to deepen my interest in studying type 1 diabetes and taking care of patients and families affected by type 1. Our group here at the University of Florida is internationally known for our work in efforts to predict and ultimately we hope to prevent and reverse type 1 diabetes using immunotherapeutics. And the one that's most relevant to SAB Bio is a drug called anti-thymocyte globulin, or known by its trade name, Thymoglobulin. That is a drug that's been around for many years and is used primarily to treat people who need organ transplants, like a kidney transplant, or who have organ transplant rejection. And through a number of studies, which I think just really reflect our group's interest in working from the bench to animal studies to the bedside of patients and back and forth as need be, our group helped discover that a low dose of this drug, Thymoglobulin, that's sort of a Goldilocks dose. It's not too hot, it's not too cold, it's just right. Seems to be the window where we can use the drug to immunomodulate more than just immunosuppress, which is what it primarily does when it's used at high doses in transplant. And when we gave it at that immunomodulating dose in patients with recently diagnosed type 1, we were able to show first in a pilot study and then a very large confirmatory study done through the NIH-funded TrialNet, which is our biggest US network for type 1 diabetes studies. We were able to show that Thymoglobulin at this lower dose definitely preserves beta cell function in new onset patients and even reduces their hemoglobin A1C, their average glucose. So that's very promising work that's now being moved into earlier in the disease process. So now going to be used in folks who have preclinical or stage 2 type 1 diabetes, those folks who don't yet need insulin. And so that all applies to SAB because they have a really novel and excellent new type of Thymoglobulin that we're looking forward to testing very soon.
Okay, so you were using in all of that work, was it the rabbit Thymoglobulin, which we're going to dive deep into a little bit later? Was it you were using the rabbit Thymoglobulin for that, and that was before joining forces with SAB?
Correct. Yeah, all the work I just mentioned was with rabbit Thymoglobulin, which is a great drug and has been around for a long time, but also has a number of warts to it that we'd love to get rid of. And SAB's product effectively gets rid of those problems with the drug and may have some additional mechanistic advantages as well.
Gotcha. Okay. Before we dive into the treatment and all of the Thymoglobulin stuff, which I've gotten that word down after saying it like 100 times the last couple days, Sam, I believe you said in our last conversation that it wasn't until like 2023 that SAB Bio emerged as a T1D company. Tell me a little bit about SAB Bio I guess before that, and why it became, I guess, a T1D company.
Sure. So SAB Bio is and had been more so before we focused on diabetes, a platform company. So what a platform company is it has a novel technology that's proprietary that could do a lot of different things. And we were developing the platform, which is a way to make human polyclonal antibodies. The only way you can make targeted human polyclonal antibodies. You can jump in if you want me to go back and explain. But at the earlier research stage, where programs are kind of in the lab and not yet in humans, it's not that expensive yet. When you get into humans, it gets expensive. So as a platform company, we spent a lot of time looking at different therapeutic areas like infectious disease, like cancer, and diabetes, and validating that the platform could have utility and bring something to patients and doctors that you couldn't get anywhere else. So we were a platform company exploring different opportunities that included diabetes. And it was in 2023, where based on the strength of the science and based on the need in diabetes, we decided to focus our efforts there, because once the company makes the jump from early stage exploratory into clinical trials, things get much more expensive. And sort of my opinion and my experience, and several of the other management team and board of directors, thinks that a company benefits from having focus. One is we can focus all of our manpower, we can focus all of our capital, and develop real expertise in one area. And the purview of being in multiple therapeutic areas is the purview of bigger, more mature companies. That's my personal philosophy. It's debatable. And so with a lot of excitement and support from people like Dr. Haller and Breakthrough, which was formerly JDRF, we focused on diabetes. We started to really focus on it in 2023.
Okay. Now, can one of you explain to us, just in very layman terms, what is SAB Bio aiming to accomplish with its therapy?
So the non-science way we think of this is your immune system has a number of cells that it needs to fight off infections. But sometimes those cells get confused and attack self-tissue. That's what we call autoimmunity. And type 1 diabetes is an autoimmune disease where those cells that are supposed to be helping you are very determined to get rid of your beta cells and get rid of your ability to make insulin, unfortunately. From a very broad point of view, SAB is hoping that this new product that they have, this humanized ATG, can go in and retrain the immune system so that it gets rid of or down-regulates those cells that are confused, while still allowing your cells that are doing the job they need to do to keep working in your immune system, and therefore preserving your body's ability to make insulin for longer. And so we typically start those kinds of studies in patients with newly diagnosed type 1. We now call that stage 3 type 1 diabetes, because those folks have a lot of beta cell function still around, even though it's diminished. And so we can study them and see, if we give them this drug that we hope does what we're saying it's going to do, compared to folks who don't get that drug, if in fact it does preserve their beta cell function, extend their honeymoon phase of type 1 diabetes, which we often call that first part where it's not as challenging to manage as it becomes over the years, and essentially kick the can down the road so that they can maybe take on other therapies. One of the advantages of the SAB product over the rabbit thymoglobulin is that it allows us the opportunity to redose it without having to worry about one of the major side effects of thymoglobulin, which is something called serum sickness, which we can talk about.
Yeah, and with your therapy, there are a couple different ways you're hoping it could be used, right? Like either I know that there's different stages of diabetes, which I want to understand better, but you're looking at not only delaying, well, with stage 3, you're looking to, is that slow it down or cure that situation? Or is it only like delaying the onset with stage 2 and stage 3? Tell me about the different approaches,
Yeah,
I guess.
all of the above. You've got it just right. So the staging system was set up a few years ago by those of us in the field to try and help people understand that type 1 diabetes is a disease that is actually well established years, if not decades, before someone needs insulin. And so we call stage 1 type 1 diabetes the time when somebody has 2 or more autoantibodies, these markers in the blood that we can very well detect now. And so we have screening programs now for families affected with type 1, and even are starting to screen the general population for these. So once we see that somebody has 2 antibodies, we unfortunately know that with 99% assurance, they're going to progress to needing insulin at some point in their life, a traditional type 1 patient. Stage 2 type 1 diabetes is when patients have multiple antibodies and start to have some abnormal blood glucose levels that we can pick up by either randomly testing them or doing an oral glucose tolerance test or having them wear a continuous glucose monitor. Those patients typically don't need insulin yet. By definition, they don't need insulin yet. That's part of being in stage 2. But their blood sugars are not quite right. And we know that means they're progressing more rapidly to stage 3, which is the time period when we used to just call it new onset type 1, when patients would present for the first time with symptoms and needing to start insulin, and we would start them then. And we even say folks who have long-standing type 1 diabetes, years or decades out, now we call that stage 4. So that's the staging idea. The first place we generally test immunotherapies is in stage 3, purely because it's a good proof of concept group. Those patients have a lot of beta cell function around. They're usually eager to participate in studies, and we have a better opportunity of potentially showing benefit over a short period of time. We can do a study and say, we're going to look at the effect in 6 months or a year and know what happens. We ultimately want to move back to stage 2 and even stage 1 disease, but those studies take decades because we have to now take patients who are at risk for needing insulin and often follow them until they do need insulin as the endpoint. And so the best example of that is the one therapeutic that's now FDA approved for type 1 treatment, which is Teplizumab. That only happened because TrialNet performed a decade-long study to show that it did in fact delay people's progression to disease by on average 2 to 3 years. My hope, my expectation is that SAB's product will be able to be used in that same space. And we're currently in the process of trying to design faster, smarter studies that use other endpoints, metabolic endpoints, rather than waiting till somebody needs insulin to say that they either, were a responder or a non-responder to the treatment. And so while, it's not realistic for anybody today say that a single drug is going to cure type 1 diabetes, we're hopeful that therapies like SAB can be part of the mix that's going to eventually lead to meaningful delay, actual prevention of disease, and hopefully one day, a way to reverse the disease.
This episode is sponsored by Omnipod. As someone with T1D and with an incredibly active lifestyle, it's important for me not to get tied up, especially with tubes on my insulin pump. I love to go swimming, lift weights, and play kickball with friends. And with Omnipod 5, I never have to worry about tubes getting in the way. Plus, not having to disconnect helps me to stay in range during all of my favorite physical activities. That includes sexy time too. See if you're eligible for a 30-day trial by visiting omnipod.com/diabetic. On there, you'll see full safety and risk information and trial terms. That's omnipod.com/diabetic. Yeah, that's interesting what you said about Teplizumab, that it was a decade-long study. I had no idea that it was going on that long. And I know that you were involved in some way with those studies. Can you tell me a little bit about your part in that drug? And for people, some people may know it as Tzield, which is the brand name, I guess now, but
Yeah, exactly. So
teplizumab
just like
is.
Thymoglobulin is the brand name for anti-thymocyte globulin, Tzield is the brand name for teplizumab. Teplizumab is a monoclonal antibody to a marker that's on those T cells we talked about, those immune cells that are sometimes confused and attack the pancreas. And so it goes in and also immunomodulates. It depletes some of those bad guy T cells and helps the good guy T cells, work better. So in many ways, it's very similar to SAB's humanized thymoglobulin. So TrialNet committed to doing a study with that drug many years ago because it didn't really have an easily commercializable way forward, much to Sam's point about, it costs a lot of money and effort to do this. And fortunately, the National Institutes of Health, which is the place where funding comes from to support the development of 99% of our drugs. So not to be political, but please continue to promote research. We wouldn't have that drug if it weren't for NIH funding, because nobody would have picked it up and done the study. NIH, because it is taxpayer funded, was willing to commit to doing it. And so we set up this trial looking at folks who were high risk, stage 2, and gave them either teplizumab or placebo, and followed them very closely to see which ones developed type 1, stage 3, and which ones did not. And so we had to wait for enough events to occur, enough people to develop type 1 to have the statistical power to determine if the drug worked or did not work. And that whole process, because finding those patients is not easy, people don't walk around with a tag on their forehead that says, I have stage 2 type 1 diabetes, was very challenging. And so it took that long to do. Fortunately, now that we got that drug over the finish line of an FDA approval, I think other pharmaceutical companies, other investors are now very interested in this space because there is so much room for improvement. It is a first drug. It is definitely not the last drug, and it's definitely not going to be the best drug. We need to do much better than even teplizumab can do. And so that's why I'm excited to be part of these efforts to study other therapeutics like SAB's humanized ATG. So TrialNet plays a major role in this space because we can and do serve as a testing ground for doing many of the initial studies. And then if pharmaceutical companies decide that is interesting enough data, they can take it and move it forward and commercialize it. And it really is a nice partnership between taxpayer funded NIH research dollars and the real world, which is ultimately going to be getting a drug to the bedside. And for that to happen, it's got to be commercialized.
Okay, forgive me if you already explained this, but who is TrialNet again?
Oh, I apologize. TrialNet is a consortium of clinical research centers, academic research centers. In the U.S., we have affiliates in Europe and Australia as well, who are solely focused on type 1 diabetes. And what we primarily do is we screen family members of people with type 1 for those autoantibody markers I told you about. And so we try to screen 25, 35, even more thousand patients a year to identify the 1 in 20 that come from family members. It would be 1 in 300 in the general population who have autoantibodies. And once we identify those folks with autoantibodies, we then follow them very closely so that we can better understand the disease, who's going to progress, who's not going to progress, why. And so that network identifies a population of patients who can then participate in studies at stage 2. Without TrialNet, like I said, we wouldn't have ever identified any of those patients who led to the approval of teplizumab. And so TrialNet will continue to hopefully play an important role in the field as we continue to try new drugs, combine drugs, design more attractive studies, and talk about ultimately putting drugs in sequence or in series as opposed to just giving a drug once and walking away. No one is naive enough now to think that we're going to have a single smoking gun magic bullet therapy that's going to cure the disease. Most of us appreciate now that, like most other autoimmune diseases that are multifaceted and challenging, we're going to have to approach it by using multiple drugs, probably given repeatedly, and hopefully at some point in a personalized way, because we understand that type 1 diabetes is not a single disease. It's 100 diseases under one label, and different patients will need different therapies. And we hope to be able to offer those. But right now we have a single drug that's approved, so we don't have a whole lot of tools, and we need to widen the toolbox.
Wow. Yeah. Thank you for all of that. It's cool to hear about like all the work that's gone into, at least we have this one drug now, right? And it's inspiring SAB to push further and create more therapies, because you're so right. There's so many different types of type 1 and reasons for why someone has it. So tell me a little bit, as it relates to teplizumab, how does this new therapy, SAB-142, how does it differ from that? What are you hoping that SAB-142 does that maybe the other one doesn't?
Yeah, so if you pretend my fist is a T cell, let's presume it's a bad guy that's not doing what we want it to do. We want to get rid of it. Teplizumab is a heat-seeking missile for a very specific marker on that T cell. So it looks for one little spot and it binds to it, and then it does what it does to the T cell. SAB's product and Thymoglobulin is different in that it's a biologically produced therapy as opposed to a therapy that's produced in the lab. And what it does is it actually sees multiple markers on the T cell all across it. And what that does is it allows the drug to frankly have a broader ability to take out the bad guy cells that we want it to see, and be a bit sort of agnostic to a single marker on the cell that we're driving the response to. And so what we've seen using Thymoglobulin clinically is that it generates some post-treatment signals that are different from teplizumab, in ways that we think are potentially better for the patient, and different in that, again, some patients may do better with one therapeutic or the other. The big challenge of Thymoglobulin, the traditional one, is because it's made in rabbits, it causes a side effect that's not very pleasant for a lot of the patients. Thymoglobulin is made, like I mentioned, it's not made in the lab at the bench side like monoclonal antibodies are. It's made by using God's biology in a very clever way. And so we take the thymus tissue from children having chest surgery, and your thymus is where your immune system sends all of its cells to become T cells. That's why they're called T cells. They go through the thymus to be what we call educated. They're supposed to learn this tissue is Mike and this tissue is not Mike, and I need to save Mike's tissue and get rid of stuff that's not Mike. That we call that education. When those cells get miseducated, that's part of why we see autoimmunity. But what somebody very smart realized was, if I take the thymus out of a human and put it into another species, a rabbit, people have done this with horses as well, those species, not being humans, will make antibodies to all the T cells that were in the thymus that it saw. And so it is a biologic response to having been immunized with a mixture of thymus tissue. So we then collect the serum from the rabbits, and that makes the medicine. We give that back to the patient. It then goes in, binds to all those surface markers on T cells that it sees, and is able to get rid of those bad guy T cells. The problem with traditional rabbit-made Thymoglobulin is humans are not rabbits, and our immune system recognizes that. And after about 10 days of the infusion, 10 days after the infusion is complete, the infusion is given only over a couple days, which is one of the big logistical advantages potentially of Thymoglobulin over teplizumab, which is a 14-day infusion. After that 10-day window, the immune system has to get rid of those rabbit antibodies, and it causes 2 to 3 days of serum sickness, which feels like having the bad flu or COVID. People get a rash, they get swelling of their joints, they have malaise, low-grade fever. It's just not a pleasant experience, and about 70% of patients who get Thymoglobulin do get serum sickness. We haven't talked about how SAB's product is made yet, but to jump to the punchline, because it's a fully human antibody, it eliminates serum sickness entirely. We've already seen that's the case from pilot studies where they've given the drug to humans, and we see that it doesn't cause that side effect. And so because it doesn't, not only can we give it with less problems for the patient the first round, it affords us the opportunity to give it subsequent times without having to worry about increasing the risks of worse serum sickness or blocking antibodies that can diminish its efficacy. So I'm very excited about the potential for the drug because we know from thymoglobulin that it clearly works quite nicely to preserve beta cell function, but it's not as clean of a drug as we'd like it to be, and the humanized ATG is much cleaner.
Mike, how did you make the connection and when that thymoglobulin could be used in this way? Because it was being used in other ways before, which you mentioned. When was that connection made that you were like, oh, whoa, it can actually be used like this?
Yeah, I mean, I wish I could take credit for the idea, but it is not novel. It's just really, again, an example of team science and a bit of trial and error and some serendipity. But going back to one of the godfathers of our field, George Eisenbarth, who sort of developed the whole model of type 1 as an autoimmune disease, and the fact that beta cells decline over time before clinically being seen as a need for insulin. He tried some of the early versions of thymoglobulin in type 1 and again showed that it provided a clinical benefit in terms of beta cell preservation, but it was even dirtier than the current version of thymoglobulin, and the side effect profile was so bad that they kind of walked away from it. But then interest in it got sort of renewed as we had better and cleaner drugs. And so the Immune Tolerance Network, which is another NIH-funded conglomerate of trial centers that does work in other immune diseases, not just type 1, did a study with thymoglobulin at a higher dose at 6.5 milligrams per kilogram, and found that it did not benefit the patients. And so our group sort of took those sets of data and said, we really need to figure out what's going on here. And to do that, we went back to mouse models of diabetes, the non-obese diabetic mouse, and started playing with the doses of thymoglobulin, and tried to find the lowest common denominator dose that would still give us the benefits while minimizing the side effects. And once we were pretty sure of that, we made the argument to the scientific community that we should come back to the field and test this lower dose of thymoglobulin, 2.5 milligrams per kilogram. And at first, we actually suggested combining it with another drug that stimulates white blood cells to be made, called Neulasta, and did a pilot study where we gave thymoglobulin at this low dose and Neulasta at a low dose to patients, and other patients got placebo, and we followed them and saw, in fact, that the drug did work at that lower dose. TrialNet then went on to do a much larger confirmatory study to separate out, well, was it just the thymoglobulin or was it the combination of the thymoglobulin and Neulasta? And what we found was that the thymoglobulin alone was doing all the heavy lifting. In fact, the group that got thymoglobulin alone did better than the group that got the combination. And so that's how we've gotten to this low dose being effective. And there are additional studies being done, or actually already even completed. We're just waiting on the results to be revealed any day now, in Europe, where they did what was called the MELD study, which they used an adaptive trial design to study even lower doses of thymoglobulin. So 0.5 milligrams per kilogram is the loading dose that we give on day 1. We traditionally then give 2 milligrams on the second day to get that total dose. And the Europeans wanted to study if 1.25 milligrams as a total dose might not be as effective as 2.5 milligrams. So we'll have even more data in that space very soon, hopefully being reported in just the next couple months, that I think will just further bolster our support for the notion that thymoglobulin and hopefully this humanized ATG will be really effective ways to move forward.
Yeah, well, Sam, how did SAB Bio discover the ability to make this human thymoglobulin?
So it goes back to our platform and what our platform can offer. And I'll just say that there's an entire class of pharmaceuticals that are derived from plasma, whether it's a human plasma or a rabbit or a horse. And that's called immunoglobulin. That's what we get from plasma. And human plasma, humans donate their plasma for this purpose, and drugs are made from the human plasma. That's often called IVIG. And we can get these drugs from animals like rabbit and thymoglobulin. And the reason animals are used is because those animals can be vaccinated or inoculated, and that makes the IVIG have a specific purpose, a specific target. And it comes with the downsides that Mike just told you about, which it causes them serum sickness, and it can only be dosed once, and it's used pretty sparingly because of that. So our platform was a way to make human antibodies or human IgG that can be targeted, like thymoglobulin, targeting thymocytes or targeting T cells. And that was done by making a cow, which we can vaccinate or inoculate, like with human thymus, that has been genetically engineered so that its antibodies or its immunoglobulin are human. So our cows, that's our platform. That's what we developed in-house in order to make these targeted human IgG drugs, which opens up a whole new world of opportunity, which we're demonstrating in type 1 diabetes with anti-thymocyte immunoglobulin. So the cows have their genes engineered so that they don't make cow antibodies, they make human antibodies. And we can vaccinate them like we do with thymocytes, and they make anti-thymocyte immunoglobulin that is not rabbit, it's not cow, it's human. It's identical to human. And therefore, we don't need human plasma donors, which we can't really inoculate or vaccinate with unknown material like human thymocytes, which we use our cow, but we make human IgG.
How many cows are there?
At the moment, I mean, it changes. At the moment, we have about 200 cows.
Okay, and this is when you're extracting this substance, is it just like you just take out like a liquid from or is it does the cow have to die?
No, the cows are plasma donors. The cows are very well treated. The best way to think about it is a dairy. So our cows are dairy cows. They're genetically engineered. They live in a place that looks like a dairy. They're handled, treated, and fed like dairy cows. But instead of getting milked and giving milk, they donate plasma, just like a human does, with the same equipment. So the human IgG business is a big business, and all over the country, I don't know what other countries do it, but at least in the US, all over the countries in shopping plazas, people go and donate their plasma. So we don't do anything to the cows that humans don't volunteer to do.
Today's episode is sponsored by T1D Exchange. You can directly make an impact on diabetes healthcare, treatments, and technology by participating in the T1D Exchange registry. It starts with a simple survey about your life with T1D, and it only takes about 15 minutes. After that, you'll have a personal portal with ongoing T1D study and survey opportunities. Plus, some of these studies even offer compensation. Signing up with the link in the show notes helps support my channel, and it allows me to continue putting out free content. You can sign up at t1dexchange.org/diabetic or click that link in today's show notes. On this show, we've talked a lot about beta cell therapy. What would you say would make this therapy stand out ahead of therapies like that?
I think what makes SAB's polyclonal human thymoglobulin stand out is, first, that it's a fully humanized antibody. We don't have anything like that to offer patients right now. Second, the fact that it's a biologic agent and it is made by immunizing these cows with human thymocyte tissue ensures that you're getting a fully broad biologic response. And in certain situations, you might want a very targeted response. In others, you need a broad response. I think type 1, because it is so heterogeneous as a disease, is actually better suited for broader therapeutics like SAB Bio's thymoglobulin. So, those two things from a scientific point of view. From a logistical point of view, this drug can be given over 2 days in an outpatient infusion center. Right now, the only other comparator is Teplizumab is a 14-day infusion. And to be frank, the logistics of that just suck. It's really
In
hard for
14
patients.
days, is it 14 days in a row, or is it 14
days
days?
consecutively of an
Okay.
infusion is the treatment for teplizumab. It's a lot for families to do. You have to miss work, you have to miss school. There are certainly issues related to equity because not all patients and families can afford to do that. The drug is incredibly expensive, so it's not like something that anybody's going to go buying off the shelf unless their insurance companies approve it, and that requires all kinds of heavy lifting from folks like me and our team. So it's just another one of the reasons we need other options in the field to offer. And so I think SAB offers both biological advantages and logistic advantages, and may provide different responses to different patients that will allow us to have the opportunity to be more personalized in our approach. And then the ability to redose is a big option. We have not historically had therapies that worked to preserve beta cell function. We now have a growing list of drugs. There's 9 or 10 drugs that have been successful in those studies in new onset patients. But they've all involved giving a therapy for a period of time and then walking away. Well, it's not very desirable from a patient's point of view. If you developed a drug and said, well, it worked, most patients and families want to keep taking it or have access to it again. And the SAB product is the kind of drug that you could very easily give, once every 6 months or once a year or once when the immune system demonstrates that it's, misbehaving again. And so I think that opportunity is a really big one that we hope to have down the road. We first have to demonstrate that it works after, the first course, but given the existing data from uses with ATG, it's hard to imagine that it won't work as well, if not better, than what we've shown in the past.
Yeah. And are there known risks to this therapy? Or, we hear a lot about immunosuppression being needed with these beta cell therapies. How does this relate to that? This was used for transplants, right? So is this...
Yeah, thymoglobulin is absolutely a transplant drug. That's what it's FDA approved for in treating transplant rejection. But the old saying that the dose makes the medicine or the dose makes the poison is certainly true here. When you have an organ transplant and without it you're going to die, you are willing to assume some significant risk, including long-term immunosuppression. Type 1 diabetes is a terrible diagnosis, but it's not a cancer diagnosis, and so the risk-benefit is a different balance. At the low dose of thymoglobulin, the time when somebody is immunosuppressed is actually quite short-lived. It's on the order of weeks. Similar with teplizumab, they're both immunosuppressive drugs. There's no way to say that they're not. They just aren't for very long, and there aren't very severe immunosuppression. So all things being leveled, you can have 0 lymphocyte counts, or you can have 500 or 1,000 lymphocyte counts, and those are very different things, even though somebody might call them both slightly low.
Okay, so this isn't a long-term immunosuppression. It's short-term. So you would get this therapy
the
maybe...
better way to think of it is a short-term immunosuppression with a long-term immunomodulation. And again, there the emphasis is on the ability to retrain the immune system so that those T cells that were previously active to attack beta cells are no longer doing that, while the rest of the immune system is still able to do its job.
And you said that there's the ability with this therapy for it to not just be this one-time thing. You get a treatment and then later on you could potentially get another treatment. Like how would that look?
Yeah, exactly. So right now with traditional rabbit thymoglobulin, if I give it to you a second or third time, the likelihood of serum sickness goes up and the severity of that serum sickness goes up. So while it works quite nicely and I would love to be able to give it again, I've been very hesitant to do that, and we have not done that yet in any of our studies or with any of our patients that we've treated clinically. Because the SAB product is a fully human antibody, we don't have to worry about that at all. And we only need to worry ourselves with when should we redose it. And I think that's a really important question that the field is still trying to grapple with and decide. And the first couple times we do these studies will probably be somewhat arbitrary, with either a time post the infusion or some signal from the immune system that says we probably need to act again, and talk about giving it, a second or third time. But I think we'll move into a time in the type 1 space where we accept that combination therapies are just going to be the norm, and we'll want drugs like Teplizumab or SAB Bio's Thymoglobulin to be used as what we would call induction agents. And then we would have maybe other therapies that we use as maintenance agents that are maybe oral and not so challenging to get and don't have nearly as many side effects as something like Teplizumab or rabbit Thymoglobulin have. And then only if and when we need to use our big punch tickets, we bring those back out and we redose with SAB Bio's antibodies or with Teplizumab or something similar.
And with these drugs, whether it be Teplizumab or this one, there is a minimum of beta cell activity needed in order for these to work, right? This can't be, I think you said stage 4 is when there's no
so if you
beta
were
cell.
a true stage 4 patient and you had no beta cell mass at all, and we measure that by something called C-peptide in the blood. So when you make insulin from your pancreas, it has a part of it that gets chopped off called C-peptide. Insulin that you take through a needle and syringe or your pump does not have C-peptide. So when we can measure it in the blood, we know that it means your pancreas is still kicking out something. If it's truly zero, then a therapy like Teplizumab or Thymoglobulin or SAB Bio's antibodies is not going to be helpful at face value. But as the folks who are developing cell therapies get better and better at them, and we have ways to replace beta cells, we're going to need these therapies to continue to keep the immune system from attacking those cells. The dirty little secret is, even if we make all the cells in the world, until we treat the autoimmunity, our immune system is incredibly good at doing its job, even if that job is not the job we want it to do. And people who've lived with diabetes for 25, 35, 40 years get transplanted, for example. Not only do they have to deal with what we call alloimmunity, the fact that they've got transplanted from somebody who isn't them, their immune system certainly recognizes that. The autoimmunity, the old memory that, hey, my immune system just doesn't like beta cells, comes right back and tries to kill them. And so for that reason, we're always going to need these kinds of therapeutics. Now, sure, people are working on ways to hide the cells from the immune system, either through physical membrane barriers or through different markers on the top of the cells. And I think those things will continue to evolve. There's still likely to be a need for these drugs, even when we start talking about treating patients with long-standing disease.
Gotcha. And I know there is a little girl in Florida that fits into this whole story, that she received a treatment. Can you just tell me a little bit about that and I guess why or how you were able to treat her with this?
With rabbit Thymoglobulin? Yeah. So, I'm a research clinician, scientist. I really like to do things using scientific method. And so, we did a clinical trial with a placebo group in our initial pilot, and it was successful. And that led to the larger trial that TrialNet did. But after completing those 2 trials, people started to ask me, well, why can't we just give rabbit Thymoglobulin clinically? It's available. It's actually not particularly expensive. And the problem I would respond back was, well, it's not FDA labeled for this. And, we don't have the most robust data in the world to support it. But after a while, I sort of convinced myself, and patients and families who were very eager for these therapies convinced me that, well, you don't do the research studies just to have the paper published. You do them to change clinical care and to make things better and to offer people alternatives. And so ultimately, I was convinced that this family who has a dad with type 1 and a daughter with multiple antibodies, they were very well informed, and a mom who was a nurse. They knew what they were getting into if they wanted to approach this from a clinical point of view, not a research study point of view. And so she was one of the first patients we treated with rabbit Thymoglobulin in stage 2, trying to do something analogous to the Teplizumab study, and she is now 4 years post-treatment and still is off insulin.
How were you able to do that? Was that just like a legal thing to do, or was she part of a trial?
Yeah,
Like,
great
you know,
question. So it was new territory for me too. So I involved our hospital ethics committee. I involved our hospital institutional review board, the folks who review our studies, and said, this patient and family wants to receive this therapy. It's not standard of care. It's not FDA approved for this. Now, keep in mind, half of what we do in pediatrics doesn't have an FDA label. It's just become the standard of care. So that is not in and of itself a reason not to do something. And they all agreed, if this was being done not because you were trying to ask a research question of does this therapy work or not, but you believe it's the best thing for this patient to do, then you can absolutely proceed and do it as long as they are, been well consented on a clinical consent form. And in fact, that's what we did. I assumed we would be doing that as a one and done kind of thing. But fast forward, 3 or 4 years, we've treated 35 people now with rabbit thymoglobulin in this way. And while people have done incredibly well, the majority of those folks get serum sickness, and the majority of those folks want another round of therapy. And I would like a cleaner version of the drug, and I would like to be able to offer them that. And that's what I think SAB brings to the table. But now we're earlier in the process. We've got to go do that study and get the right data so I can feel comfortable do it. I'm certainly not going to just jump into giving patients the new drug, but I want to move quickly towards being able to do that, and that'll require a clinical trial.
Yeah, that was great information, and I appreciate you giving me that context, because that leads perfectly into where I want to go next, which is the trials that have already happened, or the trial. So, Sam, tell me a little bit about your phase 1 trial, which you completed. How long did that take, and what were the findings that came out of that?
It took about a year from when we started enrolling subjects, volunteers, to when we had meaningful data that we could share with the public. That's the time it takes to get the patients in and then follow them for a certain period of time. And what I'm referring to is following them for 120 days and collecting data. What we learned in that trial, the two most important things, is that the patients did not get serum sickness, and that the drug behaved like thymoglobulin, or the rabbit drug. So we can be pretty confident that we have a drug that does what thymoglobulin does, but doesn't cause serum sickness and can be redosed. So we're thrilled. We're really excited, and I think that the community that Dr. Haller works in that treats these patients are equally excited to start studying it more and seeing results in their own patients.
How many people were involved? And I know that the people involved were healthy individuals. They didn't have type 1. So kind of explain that to me.
Yeah, the objectives of this phase 1 study, to show that the drug did not cause serum sickness and to show that the drug behaved like thymoglobulin, did not require patients. We could do that in normal, healthy volunteers. So we did, because that's faster, because they're easier to find, so you can do it faster. But now we're going into patients. We have started, and we will continue to.
Yeah, well, that's with phase 2B. So what are your next major clinical milestones that you're trying to tackle with this phase?
So now in patients, we want to prove definitively that the drug is effective at preserving C-peptide, and possibly even showing that in addition to that, it helps patients with their glycemic control, or control of their blood sugar. So, but first and foremost, we want to show that we're preserving C-peptide, which is preserving those beta cells that those patients have when they start the study.
How long will this phase be? Like, how much bigger is this phase than phase 1?
So phase 1, we had approximately 50 adult healthy volunteers. And in phase 2B, we want to have about 140 patients, type 1 diabetes patients. So it's more than twice the size, but it's also in patients which are harder to find. And we think it'll take about a year to find all the patients we need, those 140 patients, and then we want to follow them for a year. So another year after that. So it's basically a 2-year study.
Okay, gotcha. And where is this study going to take place?
We plan on opening centers, doctors' offices that treat these patients, and also are do some level of research all over the world. So Australia, Europe, the UK, and the US.
I know that you measure your beta cell level with a C-peptide test. Do you happen to, like, off the top of your mind, know what that level kind of needs to be? And I assume this is only stage 3 patients that for this next phase, right?
Yeah. When we get into those clinical questions, I'll let Mike answer because he's the expert.
Okay.
So the new onset stage 3 studies historically have limited enrollment to patients who are within 100 days of that diagnosis. And as much as I frankly hate that requirement, we're sort of stuck with it to be comparable to prior work. So that seems to continue to be in our world. But what I think is more relevant is, in fact, that C-peptide value. And what all of our studies have used in the past is they have to have a C-peptide of at least 0.2 picomoles or 0.2 nanograms, depending on which units you're using, when we screen them to see if they're eligible. The good news is the overwhelming majority of newly diagnosed patients have C-peptides well in excess of that. There's very few who actually screen fail just because they don't make enough beta cell function, which speaks to the potential for these drugs to be helpful to those patients. But my long-term hope is that we can someday move away from that because the day you get diagnosed is fairly arbitrary. Some people get diagnosed the minute they have a tiny little symptom, they pee one time extra. Other folks come in full diabetic ketoacidosis, but we call that the same day of diagnosis, even though the disease is very clearly in different sort of states. So I'd rather we move towards C-peptide entry entirely and get rid of the time from diagnosis, but the field's not quite there yet.
Yeah, I mean, I myself, for 2 years before diagnosis, was peeing way too often, and I just didn't catch it. But I think that this is why, another reason why awareness of type 1, the symptoms, what it could look like in stage 1, 2, 3, why it's so important to get the word out. So people listening, I hope that you just see like how much power there is to getting the awareness out, because also you could prevent someone potentially from developing it. And there's great work out there to help those people. So kind of if someone listening right now wanted to get involved in this study, this upcoming study, are you still looking for people for that? Like, where could they go, I guess, to learn more about it?
So we haven't started looking yet. We're getting ready to start, and we hope to be opening sites starting in June. And they don't all open at once, so they kind of open in a staggered fashion, and also by jurisdiction because of the filing with the different regulatory agencies. But the first thing to do is ask their endocrinologist. I mean, if they've been diagnosed with type 1 diabetes, they have an endocrinologist, right? Or their family doctor, if they don't, should ask about it. And I imagine there'll be a website up that they can be pointed to. If they don't have either of those things, they can stay focused on our website if they want, SAB Bio, and we should point everybody in the right direction. But really, a discussion with their physician is the first step.
And with phase 2B, that is stage 3 that you're looking at, right? And what would come after phase 2B? Let's say it's not, you don't get the results that you want. Does that mean that stage 2 is out of the question, or is stage 2 still a direction that can be looked at if stage 3 isn't successful? Let's start with that.
We need to see something that encourages us. What we'd like to see is a very definitive answer, but we'll learn a lot. right? We'll have a lot of data that we've collected in 140 patients, and we'll learn a lot. And we'll take that result, and it's what you call make a data-driven decision about what's next. I would like to think we'll have enough information to go into phase 2, and in reality, we're probably going to be in stage 2 patients before we have the results from the stage 3, because we want to be studying this in as many different types of patients as we can.
Now, Mike, what about you?
From a science sort of point of view, we learn something from every study we do, even if the primary endpoint isn't met. And just because a study's primary endpoint doesn't meet statistical significance does not mean that a drug doesn't have significant benefit. We might just be using it in the wrong way, in the wrong population, at the wrong time. So, I expect this drug's going to work in stage 3. But if we get some other result, we may learn from all the other studies we do in terms of understanding the immunology and how it works, that it makes sense to try it in stage 2 or even stage 1 and have a different result. We've seen that before with other drugs where they've worked in one stage and not another, and vice versa. So a negative result is not the end from a scientific point of view, but it certainly makes it challenging from the commercial point of view. In this particular case, though, this is as de-risked as something could be, because we've already shown with rabbit thymoglobulin that it works in 2 studies and in clinical use. We've seen in the laboratory that the new SAB product does the same things that rabbit thymoglobulin does, and some others even better. And we know from giving it to healthy humans that it doesn't cause serum sickness. So in my book, it checks all the reasons why I'm super excited to get the study done in stage 3 patients. Hope that we see a positive result, and then be able to offer it to other patients in other stages, and perhaps second rounds and in combinations with other drugs.
You mentioned this earlier, but I really do want to ask you directly. Why should people in the T1D community care about funding for the NIH, the National Institute of Health?
Pretty much everything we know about the disease and how to improve outcomes for patients and families affected by type 1 has come through scientific discovery, much of which has been funded by the National Institutes of Health, and specifically one of the branches that's called the NIDDK, which involves diabetes, kidneys, and digestive diseases. The NIDDK is the specific subgroup of the NIH that funds TrialNet. It funds the Immune Tolerance Network. It funds large studies in type 1 diabetes follow-up that have helped us understand, why people get complications and why they don't, and why we should treat, why glycemic control matters, why we've developed continuous glucose monitors. And so without that funding, in partnership with the commercial world, we're not going to ever see the things that people are hoping for, which is a cure and better treatments until that cure comes, and hopefully one day a way to prevent the disease.
Well, in a more tangible way, though, for people listening, how is their treatment today that they're receiving, whether it be technology or medication, what are they using what are patients using today that directly came from NIH funding?
Well, the most obvious example that applies to this conversation is Teplizumab. Like I talked about in the past there, without the NIH and TrialNet doing that study, that drug would have been shelved. The backstory is a number of companies had developed it and shown that it preserves C-peptide in new onsets. But there was a challenge in convincing the FDA to give it a label, and without a label, you can't commercialize a drug and can't sell it. And so the way the label came about was because the FDA felt that a delay of disease, a prevention for some time, was enough of a clinical outcome to warrant the drug getting that label. Without TrialNet, without the NIH, that drug doesn't exist. We're not talking about Teplizumab. We're not talking about Tzield, because it would never have gotten that name. And it doesn't encourage other pharmaceutical companies and investors to come into the field. And so I think because of the NIH and because of that experience, people today are able to get that treatment. And several hundred people have been treated with Teplizumab as patients, not in clinical trials. 4 or 5 times the number of people that were treated in that clinical trial that was used to get the drug licensed have now already been treated with the drug after that. So that's just one example of how care is directly affected by those dollars.
Yeah, and I just hear this family member in my head saying, "Well, why don't we let the rest of the world do it? Why is the NIH specifically important in this? Why can't the rest of the world and their organizations do it?" I'm sure many people are like, "Yeah, let them do it. We've done enough."
Well, I think the reality is the many places in the world don't have the same expertise and don't have the network set up to do it and don't have the wherewithal or the desire or the capital that it's even a possibility. But, as many problems as we have in the United States, we are blessed to be in an incredible country that can do these things if we choose to do them. It's just a matter of determining where we're going to put our resources. And so I would come back to say, if you live with type 1 diabetes and you want things to be better for you or your family, you need to support this kind of work because it's not going to get done. Not everything has a direct, profit-driven, company-driven line. Sometimes you just have to do the science for the sake of science. And I haven't even spoken about the basic science work. All the stuff that happens in the lab before something comes to me to be able to even offer to a patient in a research study, let alone a commercially available drug. Almost all that work is funded by the NIH. And if it doesn't happen, teplizumab doesn't ever get developed into existence at all, let alone get commercialized. So you need the whole process. You need everybody funded along the way. You need young investigators, who are kept in science. If we destroy that pipeline, all those opportunities for improving outcomes at the end of the day are going to go away, and patients are going to suffer.
Yeah, and I mean, I'm almost thinking of it as, our taxes are like this GoFundMe for the NIH, which can better its people, our society. We could be healthier. We could treat these diseases or delay them, right? So this isn't it. Our government should be investing in our health, and we should want our dollars to go towards something like that.
And just to add to that, when you use the term investment, the return on investment is a no-brainer. For every dollar of NIH funding that goes into research, it generates $4 or $5 of economic output for the community. And similarly, for all of these dollars spent on discovery, the health savings from improved outcomes is in the trillions of dollars. So, it's not fair to look at one side of the expense sheet. You got to look at expenses and you got to look at income. And so when you look at it in a fair way, we get a very good deal for the money that we invest, and we have to keep doing it if we want things to continue to improve.
Yeah, I have one last question for both of you, and this is hot off the press. Vertex announced that their Vertex 264 therapy, which wouldn't require immunosuppression, failed, and that they're ending that trial. I'm just curious to hear if either of you have a perspective on that.
I do. I mean, certainly it's disappointing. We'd all love to have cell therapy replacements that didn't require organ donors and that didn't require immunosuppression, but the science clearly just isn't there yet. I think it speaks to the need for the kind of drugs that we're talking about today continuing to be developed and improved. My hope is that we can combine those two things one day, cell therapy and immune therapy, in a way that's palatable, tolerable for patients, and allows them to not have to take insulin every day. So it's it hurts to see that study come out and be negative. I don't think it's particularly surprising because it was a really high bar to ask of a therapeutic to do that. And I hope that it will not discourage the field from continuing to explore that space, because I do think it's important, and it's an important way forward for all the people out there living with stage 4 type 1, which of course is the majority of people with type 1. If you think about it, 99% of the people that you walk around and meet who have type 1 are not newly diagnosed. And so as important as immunotherapies are for newly diagnosed patients and for folks who we can delay the disease in, we are ultimately going to have some way to regenerate beta cells. And so I think they're going to ultimately work in concert.
Yeah, but over time, and as awareness grows or we get ahead of diagnoses, therapies like SAB Bio, and others we've discussed, those we may never get to stage 4. You know, I mean, that's the goal
From your
here,
lips to
right?
God's ears, you know, if only
Let's
we
just do
had
it.
effective therapies that prevent the disease, we wouldn't need cell replacement. And absolutely, you know, the old adage of an ounce of prevention is worth a pound of cure is part of the reason I am interested in the immunology of type 1 and efforts to prevent the disease. But we still have a long ways to go. You know, as one of my colleagues said, we're probably at the end of the beginning. You know, we finally have gotten across the threshold where we have a therapy that meaningfully does something enough that the FDA has given it a license. But hopefully there's many more to follow, and we'll keep doing better and better.
Well, I want to thank both of you for coming on the show. This was so interesting, and we talked about some complicated stuff, and I think that together we all really will help people understand what is going on. So thank you for clearing things up for me, and I'm excited to see where things go, and hopefully we eliminate stage 4.
Amen.
Now, what do you think about SAB Bio's work and their therapy? Let us know in the comments. And if you want to learn more about different therapies and treatments or research for a cure, we did a full podcast episode going through all of the ones in the works with Ginger Vieira. That I'll put in the show notes, as well as Cerenova. We had them on the show and they talked about their cell pouch for type 1. So you can learn about those with the links in the show notes. And for even more, make sure you head over to diabetech.info, where we've got articles coming out every week. We even wrote about some of the cures we've talked about, so you can learn more over there. I'm Justin, and I'll see you next week.